Hello and welcome to this broadcast of ACS Science Talks, connecting the world through science. This is the virtual lecture series, Scientific Talks by Specialists on Specialized Topics for a Specialized Audience. I'm Kunal Gupta and I'm pleased to be your host for today's broadcast of ACS Science Talks. Before we begin the session, it is my pleasure to invite Dr. Diksha Gupta, Director, Global Strategy for Society Programs at American Chemical Society, for an opening remark. Diksha, over to you.
Thank you, Kunal. I'm pleased to welcome all of you to this broadcast of ACS Science Talks, a platform by ACS to connect the global scientific community. As the Director of Global Strategy for Society Programs at ACS, I'm honored to interact with you today and share our global outlook and mission to support all science professionals. At ACS, we are dedicated to provide the necessary resources for students, scientists, academicians, industry professionals, and broader scientific community to thrive in the dynamic world. Our focus is on fostering innovation and effectiveness in scientific education. We provide access to opportunities, resources, skills training, and networks, ensuring that you have the support you need to excel in your endeavors. By doing so, we contribute to the growth and success of the global scientific enterprise. We believe that diversity, equity, inclusion, and respect are fundamental values that drive progress. We are committed to promoting these principles as we work to identify and dismantle barriers to success. Our aim is to create a welcoming and supportive environment for all individuals, regardless of their background and circumstances. Together, we have the power to bring a positive change. Through collaboration, knowledge sharing, and the application of scientific expertise, we can address the most pressing challenges being faced by humanity. By supporting science professionals like yourselves, we are laying the foundation for a brighter and more sustainable future. I'm grateful to each and every one of you for your dedication to the scientific enterprise. Your work is essential, and it is our privilege to stand alongside you in this remarkable journey. Thank you, and I'm sure you will have a great time listening, Professor Lim. Over to you, Kunal.
Thank you so much, Diksha, for that wonderful remark. Our speaker for today is Professor JitKang Lim. Professor Lim is at the Department of Chemical Engineering at Universiti Sains Malaysia, or Science University of Malaysia. He holds a PhD in Chemical Engineering from Carnegie Mellon University. He is a distinguished researcher in the field of magnetic nanoparticles and has made significant contributions to the development of the low-gradient magnetic separation process. His expertise has earned him prestigious awards such as the Takeda Young Entrepreneurship Award and the National Young Scientist Award. The National Young Scientist Award is one of the highest awards that is given to a young researcher or young scientist under the age of 35 years in Malaysia. Professor Lim is actively involved in international collaborations. He also holds a courtesy appointment at the visiting research professor of physics at Carnegie Mellon University, and has recently been appointed as the China ASEAN Scholar of Tianjin University. He has secured funding from various national and international agencies for his research projects, focusing on colloidal and transport behaviors of magnetic nanoparticles. In addition to his academic pursuits, Professor Lim is committed to promoting green technology and was awarded the Green Penang 3A Certificate of Appreciation in 2013 from the Penang Green Council. As a founding chair of American Chemical Society local chapter in Malaysia, Professor Lim plays an active role in fostering scientific collaboration and knowledge exchange. He is also an editorial advisory board member for Langmuir, a leading journal on interfacial sciences published by the American Chemical Society. Professor Lim's passion for research, dedication to teaching, and outstanding achievements in this field have solidified his position as a respected academic and researcher in Malaysia and beyond. So thank you, Professor Lim, for joining us. The stage is all yours.
All right. Thank you so much, Dr. Kunal Gupta, for his kind introductions. Before I start my presentation today, I would like to thank the ACS India under the leadership of Dr. Disha Gupta and her team for all the coordination efforts and time they all put in to make this event a success. And today I would like to share with you, let me share my slides. Today I would like to share with you some of my research findings which is based on the long-term collaborative work which I've been pursuing with Dr. Derek Chan from Universiti Sains Malaysia and Dr. Chun Chao Liu from Chinese Academy of Science on magnetofluoritic separation of the microalgae. So firstly, I'll talk mainly about the environmental engineering applications of magnetic separations. Then I'll use it as a stepping stone to move into the magnetotactic micro-robot construction that later I'll bring out the transport behavior of it and eventually the challenges associated with engineering applicability. All right, the starting point, even though the research title so fancy, right, is moving in Nanoworld, but the starting point is not from in the Nanoworld. In fact, when we first started to come across a problem is actually in 2010, in which my colleague, he referred to me where our USN campus is here somewhere in the red circles, and there's quite a number of freshwater fish farm just very close to our campus. And what the fish farmer doing here is actually they feeding the fish and with high nutrient fish fat, and eventually this nutrient accumulated in the fish farm water and promoting the growth of microalgae. And so one of the solution practiced by local farmer is actually they just mix this water that's contaminated by microalgae with rainwater, and eventually they just channel it out into the nearby river. So from their Google map image alone, if you see it, the whole river actually is quite green. It's because microalgae start to bloom in the river area, and there's a lot more fish farm here. So if you take the samples of this water from the river around, right? So you can see the water is very green and it's heavily polluted with microalgae. So the microbe is actually growing quite rapidly inside the water. So our take on this, of course, is a bad news for the local community, especially our campus is so close to the fish farm here around. And our hypothesis is, even though the microalgae cell is a biological cell, of course, most of biological cell is non-magnetic. If we have a way to attach the magnetic nanoparticles onto the microalgae cell, then all we need is to apply an external magnetic field to collect the microalgae cell out from the aqueous environment. So the whole idea is very straightforward. As long as we have a way to impart magnetic properties towards the biological cell, then we can separate them out by using magnetic field. So of course, the first question is how to promote such a particle attachment. Because if you really look at it, the cell, right, the microalgae cell is mostly negative. And the magnetic particles that we want to use, which is safe and can be used for environmental engineering application, is mostly iron oxide nanoparticles, and iron oxide nanoparticles is also charged negative. So if you just mix them together, they experience electrostatic repulsions, and the particles is not going to attach. And so if you just mix the particles together with the microalgae, and if you try to separate them, and this is what is going to happen, you have the microalgae cell remain in the water, and whatever you add in, you have any consequences of the time, any impact towards the separation. And since the whole interacting scheme, right, is driven by electrostatic repulsion. So as long as we have a way to actually reverse such electrostatic repulsions, we could actually promote the particle attachment. The easiest way is actually the coated particles with a kind of charged molecule or molecule that carries some kind of charge, and it have to be cationic, which is positively charged. And so we try to decorate the particles with the polycation is a polydieryl dimethyl ammonium and PDDA molecules, and it's charged positive. And eventually we manage to promote the attachment of particles. And we can also do certain kind of interaction calculation. That means we calculate the interaction potentials between the particles and cell by making certain kind of simplified geometries, and based on a classical theory developed by 4 known scientists called Diagon, Landau, Verwey, and Overbit. So it's based on DLVO theory. And we managed to construct the interacting potential between the particles and the cell. And by doing that, we could actually predict, not only experimentally make sure the particles can attach onto the cell, but theoretically we could predict whether our surface functionalization scheme can actually work or not. And so far, so good. For the freshwater samples, by using Corora as one of the model system, my student, Pei, managed to actually prove by surface functionalizing the particles with PDDA, we could actually promote the separations up to about 98 something %. And the theoretical predictions of this DLVO theory, by taking account of only van der Waals interactions and electrostatic interactions, is worked perfectly. The particles attachment is exactly as what we have predicted. They managed to attach. And of course, then once you got it, we declare the victory and we publish the papers, and we say that the whole interaction scheme is actually driven mainly by electrostatic interactions. Later on, once we shift our attention to seawater species, because microalgae is not only appear in the river, it also can appear in the seas. So we want to actually expand the applications for not only separating the microalgae in the freshwater conditions, but also in seawater conditions. And actually our strategy failed, and it failed very miserably. And this is kind of counterintuitive, because if you really look at it, right, the seawater is full with free ionic species. And this ionic species actually provides something called day-by screening. That means if you have a negative particles and the positive particles, that's why you have a negative cell and positive particles, right? If you have a lot of free ionic species around, you will screen the interactions between them. So in fact, you really suppress everything and making the interaction much more stronger. However, this is not what we have observed. In fact, if we mix them together, the bare iron oxide particles and with the surface functionalization ion particles, we got the same outcome, which is 0% separations. So the whole thing is this: how could this happen, right? But our experiment show it, right? So if you really look at it, right, by using the classical DLVO theory that I described just now, the whole thing is very successful, right? It can predict the freshwater very nicely, but in seawater conditions, it's actually predict attraction, which we didn't see in our experiments. And so something is seriously missing in this kind of model. So what had been missing? So after some studies and things like that, we realized one thing is we are missing off one key interaction between the particles and the cell, which is called Lewis acid-base interaction. By incorporating Lewis acid-base interactions into the total in the entire model, and by repeating the calculations, now our mathematical model can actually be more than successful enough to predict in which under sea conditions they have a huge repulsions, mainly because of Lewis acid-base interactions. And this is actually preventing the iron oxide particles, even though it's coded with a PDDA, even though they're supposed to be electrostatically attracted to each other, but because of Lewis acid-base interaction is so strong, actually it prevent the attraction to be happenings. And so in fact, actually we published another paper to talk about it, but surprisingly, there's still people, keep citing our old works and keep talking about the electrostatic interaction is actually one of the main driven phenomena. So that is only about the particle attachment. So once we manage to attach the particles onto the cell, right? And if we expose it we introduce a magnetic field, then we could see the microalgae after mixed with particles, and we can manage to separate them out quite rapidly. And eventually, if you look at it, the water will turn into crystal clear. In fact, we managed to separate most of the microalgae out from the solution. And if we really look at it into the transmission electron microscope images of the particles, and we could see the successful attachment of particles onto the cell. And actually, these results, right, actually reveal to us two possibilities. The first possibility is straightforward. By surface functionalized iron oxide particles, we could impart the magnetic properties onto the cell, and that's why we can separate them by using magnetic field. The second one is, in addition to unidirectional separation, that means we can only separate them in one direction. If we really think about it, right, if the part of the particles can attach to the cell, we now could rely on these magnetic particles to guide the motion of the cell, right? Which means we could really move the cell by using a magnetic field. And in fact, this can lead into a very interesting robotic kind or micro robot kind of design. And actually, we are not the first one to propose this. Before us, there's so many groups that coming out with a very fantastic design about combining artificial substrate with a microstreamer. Here, the artificial substrate, for example, if it is a magnetic nanoparticles, then it could impart magnetic field onto the swimming cell, and eventually leading to the active control by using magnetic field, so we can control the motion. And in addition, this active substrate can also serve as a cargo, so we can use this cell to pick up this active substrate, and eventually, if we make it magnetic, so in addition of it to become a cargo, we can rely on the magnetic field to actually guide the motions of the entire constructs. That means we have a way to control the motions of the microbot. And in fact, right, there's so many kind of applications have been proposed by using microrobot, and they are mainly used for biomedical applications, and some of them have been used for diagnosis purposes in microfluid channels, and some is even used to study fundamental questions such as the cellular functionalities of certain kind of cell. However, there's one caveat. The caveat is this: when we think about to attach magnetic particles onto the swimming cell, right? And we go and search in the literatures, there's a lot of ideas there, and people using all kinds of different cells. For example, here, there's so many different kinds of cells. They're combining with the magnetic bits, which is not nanoparticles anymore, it's micron-sized particles of all sizes. And eventually, they all reported they managed to control the motion of particles and altered the average moving motion speeds, the moving speed of the particle, or of this construct, which is the microbot constructs. And if you really look at it, like all these studies, most of the average speeds been reported so far is from few micrometer per second until about 10 micrometer per second. These speeds are actually caught our attention because almost at the same time, we managed to track iron oxide magnetic nanoparticles, which is roughly about 35 nanometer nanoparticles. So there are 35 nanometers, which is way smaller than 6 micrometers or 10 micrometer. And the average speed which can be achieved by these 35 nanometer iron oxide nanoparticles is roughly about 10 micrometer per second. So they're at the same order of magnitude, right? The rest of this. And why this actually causing trouble? Because if you really think about it, right, the magnetophotic forces is actually proportional, is defined by this equation, is actually proportional to the Vmax, in which Vmax is a magnetic volume of the particles that you use. So in other words, the magnetophotic forces is directly proportional to magnetic volume. If that's the case, if you compare the 35 nanometer particles and a 6 micrometer magnetic bits, for example, these cases, even with only 10% of the volume of these bits is magnetic, 90% the rest is not non-magnetic, we are still talking about 500 times difference comparing to small iron oxide nanoparticles. So by right, we should witness the speeds of 500 times much larger in this construct, and actually we didn't, right? So it's very consistent. Everybody report with the speed of roughly about 10 micrometer. So which is on the scale of a nanometer size particles. So this actually caught our attention. So we decided to pursue it and really try to answer these questions. And my student, Wei Ming, actually managed to propose an idea. What he proposed is we could construct a microbot, learn from our experience of dealing with microalgae, like what I explained to you all just now, which is the starting point of this project. So though we can construct by attaching using the same surface functionalization strategy that we developed, we could attach magnetic particles onto a microalgae cell. And the model system that we're using is Chlamydomonas rihardi, which is a single green algae cell with two flagella and at the front of it, or at the anterior part of it. And please pay attention on this. I'm going to refer to this number quite a lot. The average swimming speed, if this guy, since they have flagella, so they can swim, right? So it swims at around 100 micrometer per second. And we're using two different sizes of microbits. So eventually we try to construct a microbot construct, something like this. So these are the optical micrograph images of the microbot, in which we can see it. There's one part is the magnetic bits, and the other part is the microalgae. So we name the whole construct as microbot. Right. So here is the lifetime recording of the microbot motion before and after we turn on a magnetic field. And here we are using a mu matter to direct and localize the magnetic field into these portions of the experiments. And please pay attention on this, the microbot construct of this one. And I'm going to turn on the video, but without sound, right? So it's just video. All right. So when we turn on the magnetic field, the microbot actually fly through and move closer to the mu matter tips. So let's look at it again. Pay attention before the magnetic field turn on, it's just actually going through helical motion. And after we turn on the field, right, the whole construct actually moving over to mu matter tips, which is not surprising because we import magnetic properties onto the cell. All right. So if we track the motion path of the whole construct, right, so we track out quite a number of them, and eventually we could decompose the motion pathway into two parts. The red circle one is before the magnetic field is turned on, and the broken line is after the magnetic field is turned on. The broken line just shows the particles actually respond to magnetic field, like what we expected, and actually move over towards the mu matter tips, or toward the magnetic field. And interestingly, the cell actually gone through certain kind of helical motion. Every one of them actually gone through kind of helical motions. And if you really look at it, right, since we constructed the microbot by using 2-micron particles and also 4.5-micron particles, and you could see it, the velocity, the velocity of it actually reduced quite significantly. Remember just now the number I call you, remember 100 micrometer per second, the velocity actually reduced quite significantly, and the apparent speed become about 23 micrometers per second. And if you check on it, right, the velocity, of course, reduced, the translational velocity, and the frequency, they started to rotate. And when the bits become bigger, the translational motion actually further reduced, and the frequency actually increasing. So they rotate more rapidly. And surprisingly, when we first look at it, we thought it's a non-surprise. It's not a very surprising result because of many reasons. But when we dig further into literature review, there's so many people have made this kind of observation. However, by different groups of people, all from top university. For example, here there's a group from Harvard Chemistry, and this group is from Max Planck Institute of Germany. And they publish in top journal using the same cell line as us, Chlamydomonas rihardi, using almost similar size of microbits, magnetic bits, microbits. And what happened is this. Certain group, they report there's no change of the velocity. The change remain 100, and some is even increasing. They could observe the velocity, actually the speed actually increased. And some report the speed like us actually been reduced. So eventually, if you dig further down to this, the Proceeding Nation Academic Science table, right, it says something about the beach attached or close to the flagella, typically in pit swimming. This makes sense. Pit located further away did not, right? So of course, if remember the sail actually swim by flagella motion, right? So if you have bit as it close to flagella and block it motion, by right you should slow it down. This is purely logical, right? It's not even having any scientific proof, but we're going to prove it scientifically later. So this is non-surprise. However, they observe a very surprising result. They say, when cell with a bit bound directly between the flagella swarm at a velocity of more than 100 micrometer per second. Surprisingly, when the bits actually at the center and fix at the center and suppress the flagella, and the microbot actually can swim much further. So this actually, and this group is a very famous group. So eventually they motivate us to further pursue what is actually happening in between, right? So whatever the result I show you so far is the average result, right? So I show you how the particles swim without bits. The number I tell you is 100. Then later on, I show you after the bits attach onto cell, then how it swims. So this is not a very good experiment. This is not a very good result in order to review what's happening. The best is actually to capture the whole process of cargo picking or load picking. That means we could have one video, we managed to capture the cell actually swim independently by itself and approaching a bit of particles and eventually pick it up and swim together, right? So this is the video that I'm going to show you. Let me play it, right? So it's a very challenging experiment because two reasons. The thing is they move very fast, and actually the whole thing can be happening. The whole system is very messy, so it's very difficult to capture this. So let's look at it again, right? If first the cells swim almost translationally, when it hit the bits and pick it up, right, it start to having the helical motion again, right? So the work is this video is captured by one of my students, Xiao Zhong, and we managed to capture about 10 pairs of this interaction where the blue line indicate before the cargo picking and the green line indicate after the cargo picking, where the red dot actually indicate where the cargo picking actually happening. And you could see it in all cases, right? The cell, when it's cell alone, without having any cargo, without having any substrate, the cell actually moves quite linearly. And once it's pick up the cargo, and what really happening is it started to undergo helical motion, right, in all conditions, right, in all 10 of the experiment. So if we really try to answer why, remember the whole question is still, is it slow down and why it slow down, right? So if we really look at it, right, we could, the easiest way from the classical mechanic perspective, like what we have experienced, we all know, right? If you, even a human being, if you're carrying a load with you, you can't run as fast as before. That is just the conservation of momentum, right? So it's just so if we additional mass, you have more momentum and things like that. So we do the conservation of momentum analysis by carefully estimate what is the bits of, what is the mass of one bits, and eventually the number is just doesn't add up. And so the whole thing that the cell that we observe is slowed down, but the slowing down process is not because of added mass for sure, because the conservation of mass, the conservation of momentum just doesn't add up. And the other possibility is once we add the particles onto the microboard, the geometry of the whole construct actually change quite drastically. And this geometry change, right, leading to the fluid drag experienced by the whole construct is completely different. So since the particle, remember, the cell is very small, they swim in the low Reynolds number conditions. So by right, the drag force should dominate the momentum. In such a way, it might be the reason. So how we did it is, so instead of introducing the particles onto anterior, that means at the center of the at the anterior part or at the front part of the cell, we try to attach the bits at the bottom part of the cell. So the presence of this bit is not going to interfere with the movement of flagella at all. And so if we observe anything, the whole thing should be because of the drag alone, because of the geometry change, not because of the obstructions of the flagella. And we managed to calculate the differences in terms of the viscous drag after attaching of these microbits. Is about, the difference is about 11% increment in terms of the viscous drag. And however, if we look at the average swimming speed of this construct, right, with posterior attachment, the swimming speed is roughly reduced to half, right? So in other words, it can't answer by just having 11% of physical strength differences. It can't answer the 50% swimming speed reductions. But one thing we all notice is if we decompose, we look at the motion path again of this construct, right? We didn't see any helical motion. After it picked up the bits, actually it still moved quite translationally. And that's important. And once we rule out the mass addition, it's not the reason for it. We rule out the geometric contribution. It's not. The only left behind is a very making sense one, in which, like what suggests by other group as well, the presence of the microbits on the stroking path of the flagella can actually obstruct its motion, and this actually hinder the way of the microbot of doing work, right? So here is a video, and to show one of these bits, right, actually almost suppress one of the flagella, and the other flagella actually can still move quite freely. So if you really look at it, I hope you can see it. Actually, one of the flagella is moving, the healthy one is still moving, complete the whole cycle, and the other one, the paralyzed one, is almost not moving at all, but moving very minimally. Again, I repeat the video. One is actually moving quite rapidly, quite healthy and natural, and the other one is not, right? And we could track and onto this, and we can really confirm quite sure the other one is actually paralyzed because of the presence of particles. And remember, again, this is the motion path where we have the translational motion before the cargo picking and helical motion after cargo picking. And so what it is, if this is paralyzed and one part is paralyzed, can we predict it, right? So we do a work balance or energy balance on this cell before and after the cargo picking, and we realize the energy differences between these two scenarios is roughly about this number. Sorry, the thing is blocking me. I can't even see it. 2.63 times 10 to the power negative 17 nanometer. And we use a mathematical model proposed by James Lighthill many years ago. And what we do is we compute by assuming 50% in hindrances of the heterogeneity. That means we assume one part of it can't move. There's only one, like what I showed you just now, actually move healthily. And we managed to compute a loss work of about 2.2 times 10 to the power negative 7. And these two numbers actually is in quite a good agreement. And this actually reconfirm or reconvene us in which the reason why we observe a slowing down and loss of the work, right, is because one part of the flagella actually is suppressed of its motion, and eventually this just couldn't really drive the whole construct to move quite healthily. So that's also the reason why it's going through the kind of helical motions. All right. So since we are going out to share this with a lot of other people, so a lot of maybe a lot of them will not agree with us. So we have to be very careful about it. So we try to confirm again, to try to convince ourselves. This experiment is this. Now we have microbot construct. Microbot construct, that means one micro swimmers with one bits, right? So we use this and we try to pick up another cargo. Let's see how it works. Right, so we have one cell with bits, one bits, and it pick up another cargo. And that's exactly what happened now. Right, so if you track the motion path of this construct, right, so what we have now is, now we have the one, again, this is average value, and this one bit, and this after second bit attachment, right? First, the most obvious one is the radius of the rotation. The whole helical motion, there is radius actually increased quite a lot, right? And its velocity is actually reducing, further reducing, and its angular velocity actually also reducing, and the oscillation per second actually also reducing, right? It all makes sense because you have another loop go on to it, right? It's also cause the off centers of the center of the gravities, and eventually the whole thing is just gone through a much larger helical motion. And in other words, it's also suppressed the other healthy part. So in completely one side of it is completely polarized, almost 100%. All right. If we talk about the microbot idea, the constructions and to understand the motion of the microbot is only one part of it, right? Since if we really want to count relying on the magnetic field to achieve active control, to control the motion of the microbot, we really need to have a system that's sophisticated enough in order for us to direct its motion. So the part of this part is what I'm going to talk about is by using some, like the very simple electromagnetic calculations, we managed to construct a magnetic tweezer that could introduce magnetic field that is strong enough in order to direct the motions. And if you really look at it, right, before that, whatever I showed you just now is very simple. Just consider first generation setup, right? So we have a mu meter. So it is iron nickel alloy that we actually use to direct the magnetic field and pointing it. So right, we concentrate the holdings into the tips and we use this tip to crack the microbot. And the second generations, we are so obsessed for having the manual construction. So we try to use, instead of electromagnet, we try to use a permanent magnet because we try to do something really cheap and really workable. And eventually and to be honest, we fail miserably as usual. And then I got one of very good students coming in, Wai Hong, in which he started his picture here and he started to actually improve the design, and eventually we modify the magnetic tweezers again and again, and eventually we managed to get something. And here is a video and showing it is a quadrupole design. That means we have the electromagnet at 4 points here. The red indicate the magnet is turned off, and green indicate
Sorry to interrupt, Professor Lim. Somehow we are not able to hear you. You're not audible. Maybe it's some microphone issue.
Sure. Can you hear me now?
Yes, we can hear you now.
All right. Yes, it says my internet connection is not stable. All right, sorry again. So I repeat these slides again. This is my last slide. So afterwards, I will just summarize everything. All right. So again, the whatever here is the bright field imaging of a real-time tracking of this motion of bits by using the magnetic tweezer that I described just now. So it is a quadrupole design. That means there's 4 poles, and the red dot indicating the magnetic field from this part is turned off. And if it is green, that means the magnetic field is turned on. All right. So please pay attention onto this black dot here. This is what we try to control its motion. If we try now to turn on the magnetic field and we manage to direct its motions, and just because recently I was appointed as the China Asian Scholar of Tianjin University, so this is what is a collaboration with them. So eventually we just write a Tianjin words, because I'm thinking about to write USM 3 words it's not very challenging. So we decided to write something more complicated. So we wrote the Tianjin words to use our system to prove actually it's working, and we still gone through optimization and things like that. All right, and that's it. And let me summarize what I have here. And the thing is keep blocking my way. All right. At the first part, I'm talking about microalgae separations as a way to combat the water pollutions. And the second part, I'm talking about the microbot construct and how the cargo picking by the microswimmer is possible. And the third part is actually, I talk about the constructions of the magnetic tweezer and design, and we're still working on it, and hopefully we'll be able to share more results with you. And with this, I thanks all of my students, hardworking students. I'm just an advisor sitting in the office. All the hard work that you saw, whatever you enjoy, is you. The hard work is not me. I'm just as the advisor that's sitting in the office. And I also like to thank the funding agencies and all my collaborators. And more importantly, I'll thank you for your attention. Thank you so much.